Good evening, everyone. Am I audible to everyone?
Yes, yes, you're
Okay.
audible.
So, welcome to today's session. This talk is organized by BioRx. It's a student-run biology community built for curious minds across the life sciences. It serves as a platform where students connect, exchange ideas, and engage in meaningful scientific discussions. As part of our continuous effort to explore key concepts shaping modern biology, today's session focuses on marine biodiversity and ecological significance of one of the most overlooked groups of organisms, marine sponges. Today's session is titled Stories of Simplicity to Significance: The Hidden Power of Marine Sponges, aimed at understanding their role in ecosystem functioning, biodiversity, and environmental change. We are honored to have Dr. Anita Mary George with us. Anita Mary George is a distinguished marine biologist specializing in sponge taxonomy and coral reef ecology. She is currently working as an assistant professor at the University of Kerala, where she combines academic teaching with active research in marine biology. Her work focuses on sponge taxonomy, coral reef ecology, and the use of modern tools like metagenomics and environmental DNA to understand marine biodiversity and ecosystem dynamics. She is also associated with CSIR, National Institute of Oceanography, Goa, and is PADI certified rescue diver with extensive research experience across global marine ecosystem. Her works also includes discovery and documentation of several sponge species along the Indian coastline, significantly advancing marine biodiversity research. We are delighted to have her with us today and look forward to an insightful session. Let's warmly welcome Dr. Anita Mary George. Ma'am, over to you.
Thank you. Good evening, everyone. Um, it's a pleasure to be here. Am I audible?
Yes, ma'am.
Okay.
Yes, ma'am.
Okay. So it's always a pleasure to be here with such enthusiastic group of students. So many thanks to the organizing team, BioRx, for making this session possible. It's not easy to put a platform together with all the passionate groups and on particular topic together. So I believe they are gonna officially starting tomorrow, and congratulations to the and best wishes to the entire team. So today we will be discussing on marine sponges, and thanks Sonia for the introduction. And I hope this session will give you some practical insights and ideas where you can apply on your journey. And I encourage all you to stay curious and interactive throughout. So let's get started to the topic straight away. So let me share the screen. Okay. Can you see the screen?
Yes, ma'am.
Okay. So we'll start straight away into the stories of simplicity and significance of marine sponges. I'll put my timing. So we'll be having roughly 45 minutes to 50 minutes. Is it okay? Or you want to be shorter? You can talk. Anyone? Nobody is talking. Okay. So before I start and discuss about marine sponges, we need to know what are sponges. So is the screen fully clear, or is it something coming in between because I couldn't see? I'm seeing only my screen. So is it okay?
Ma'am, keep it in slide mode. Then it will be full screen. And you
Full.
can take as much you time deep inside.
Okay. So is the screen okay?
Yes, ma'am.
Is it
No,
something? the presentation screen is coming in bothering you. Is it so?
You need to put it in full screen mode.
It is in full screen mode.
But we
But
are seeing all the slides on the left side.
oh, okay. How can I do that? 1 second.
Corner, ma'am.
Should I share the entire screen or just the window?
Window, ma'am.
Okay. Oh, sorry. 1 second, please wait.
No issue.
Sorry, I accidentally closed. Yeah. Um, can you see the screen now?
Yes, ma'am, but the slide is not full view mode.
Oh, okay. Is it just the slideshow, right?
Yes, ma'am.
Okay, okay.
Now it's okay.
Okay. Now you can
No
see the
problem.
screen?
Now it's full view. Yes.
Okay, okay. All right. So, okay. When I ask about what are sponges, so the first thing will come into your mind is maybe the bath sponge that you see in your kitchen, or the sponge cake, or the SpongeBob that you see in the cartoons. So we need to really know the two sponges that you see are the real sponges, and this is the skeleton of it. So what happened in Mexico, like in olden days, what they have to hunt a lot of marine sponges from deep sea, and then they have to export for getting this skeleton of the sponge. But then people started doing all these synthetic sponges. So let's get soaked in the living sponges for the next 45 to 50 minutes. Okay. So when we have to know about what are sponges, early naturalists actually classified them into plants because they lack organs. They don't have any true tissues, no circulatory system, nervous system, nothing. They are very, very simple, humble group of animals, and also sedentary. They are sessile. They have to attach with some kind of rock or any kind of hard bottom, and also they have a lot of branches. So the early taxonomists, Linnaeus, they thought in plantae, they classified in sponges into plants. So only in the 18th century, sponges were classified into animals based on their body composition, elastic collagen, because the collagen is just the same as our skin. Okay. And they have central cavities, and they have very distinct water currents, which I'll show you in a while about their anatomy, how it get into their filtering and other role. So when you get into sponges, the major phylum is Porifera. So Porifera means they are pore bearers. Pori means pores and fera is bearers. So pore bearers. Well, whenever you see a sponge, you will see a lot of pores in that. So it can always have a clash with ascidians where it looks very similar, but the surface is very smooth and they don't have pores. So these guys always have a lot of pores. So why they have pores, and we'll get into that in a short while. Before that, we'll see about their evolutionary journey. So when you see about their ancient lineage, they are the primitive multicellular animals. So the fossil sponges we get is from 750 million years ago, and they fall into the Metazoa group. And these Gibbs 2018 from Max Planck University and Max Planck University research in 2019 also proved that sea sponges are probably the first animal on Earth. And this Massachusetts Institute of Technology have proved this using molecular studies. And always sponges have a clash with comb jellies. So these the research always they say who comes first, sponges or comb jellies. So when with this clash, always so many molecular studies happens and so many papers are there, but always sponges win the match, and they are the first group of animals that come to they are the early and the first animals. So when you learn about the definition about sponges, they are a very sedentary filter feeding metazoan which utilizes a single layer of flagellated cells, that is choanocytes. I'll show you what the choanocytes looks like, to pump a very unidirectional water current throughout its body. So Berquist in 1978 put forward this definition, and it is very true, and still we follow this definition. No changes at all, because they are very, very simple guys. So where can we find these sponges? Right from the tidal zones to the deepest abyss. Okay, so these are from the tidal zones. You can see they can be attached to the rocks, they can be encrusting, they can be subtidal. Even when you dive, you can see in 4 meters, 3 meters, 12 meters, even 40 meters that we can go underwater. And then we can, research shows they are in the deep sea also. So deep, even in 3,000 to even 8,000 meters, these guys are everywhere. So right from tropics to temperate zones, they are ubiquitous. Everywhere we can find them. So it's just easily, we can do a research on them very easily. So you don't have to jump into the water. You can just simply wait and just get into the water, and you can get it, these sponges even in the intertidal regions. So they are not confined only to marine. There are freshwater sponges also. So they can be found in canals, rivers, dams, and many other small pools also we can find them. So mostly freshwater sponges are this grayish or mostly greenish in color. So you can easily find them. So in this presentation, I'm just bringing in most of the pictures and informations on marine, and because freshwater sponges are a small group. And this is the choanocytes I was telling. So when you say check into their anatomy, they have a single spongose seal. The outer side is the pinacoderm, or you can see the dermis or the cortex we say, and then the internal we have all the cells called choanoderm, and these are the choanocytes with all the flagellated cells. So you can simply with a simple diagram I'll show you. This is the simple cavity, and all these spicules or the skeleton. These are the important guys for getting into the different species. But what they do with their collarbones, that is the choanocytes, or with that flagellum. So with that, it keeps
Excuse me.
so they come into the group Porifera. And when we see the evolution thing, it is coming into fossil sponges. They are the first sponges probably on the earth. And with all these clashes, with comb jellies and sponges, always sponges come to the first, because they always, with all these molecular studies, shows that sponges are the first animals. And this is a simple definition which I have gone through, because it is a sedentary filter feeding metazoan which utilizes a single layer of flagellated cells to pump a unidirectional water current through its body. So I'll show you how the sponges looks like. So where we can find them? We can find them in the intertidal region, subtidal, and the deep sea also. And we can just see them even without getting into the ocean. We can see them just by wading along the ocean. So we do have freshwater sponges also. Say normally they'll will be green in color, and as I mentioned, you can see them in rivers, dams, canals, everywhere. So in this, we'll focus more into sponge anatomy. So this is the sponge spongocele that I said, that is the internal region which full of flagellated choanocytes. These are the very interesting guys which help them, this whole simple sponges, into the mighty organism that do wonders in the ocean. So I'll show you the simple sponge feeding anatomy. Like, so this water always comes through the ostium, so it is through the sides. Through the sides always it comes into that, and it will, after filtering, all the water will go out. So this is a constant filter feeding group. So always, like, this is 24/7. They don't have any activity, just without any brain. They focus only on this. So they are the best vacuum cleaners of our ocean. And that's why we keep these sponges in aquariums to filter water. And wherever these guys are there, it will be clean water. So they keep on filtering the water all the time. And they will have the unidirectional currents of water. So many interesting characteristics are there in sponges. So we'll quickly go through, because already we wasted a little bit of time. So mostly the adults are sedentary. So mostly they have a little bit of locomotion, but mostly the adults are sedentary. The larvae are a bit motile, so they are incubated within the parent, that is viviparous, and then broadcast into the seawater to become oviparous. And these are the buds of the sponges. Like, when this is a interesting tennis ball sponge, we call. It's round, ball shaped, and these buds are always yellow in color. Very beautiful sponge. And how they reproduce, both by sexual and asexual reproduction. So this in this picture you can see the asexual reproduction by budding. I'll show you a detailed one. And with the egg cell and the sperm cell, it gets into fertilization, and embryo is formed, just like I get into a blastula form, and then get into a new sponge when they fall on a substrate. So it's a very simple type of I'll get into this asexual and sexual reproduction in detail. And their interesting capabilities, they are totipotent. So even if you take a tiny bit of sponge and put it and split it and keep it on a aquarium, they can grow into a full sponge. And also, if you take 2 or 3 different species and then just smash them and put it in water, and H.G. Wilson in 1925, he did a research on 3 different sponges and put it in a aquarium. All the species on their own, they find their group together and form into a full sponge after some time. So they are highly brainy people. Even though they don't have any brain, they behave so nicely. And so that's why when we collect sponge, we always leave the base and take only the a little, because for research we need only a tiny bit of it. They come in different colors, shapes, and size, and they lack, most of them, they lack symmetry. So we have many different types of sponges. I'm just showing you the different variations of sponges, but we will get into the real sponges like this. So we have loggerhead, boring sponge, breadcrumb sponge, giant barrel shaped sponge, mermaid's glove. These and they come in beautiful colors. That is the very interesting things that we can, when we dive and we can see them, varied group of different sponges. So this is the detailed, adult, and I thought of showing you a video, but I think sometimes if it gets stuck, so I'm not getting into that, but I'll share the link and everything if anybody is interested. So asexual reproduction can happen by 3 different methods. Fragmentation, once you split it, and then you can keep it on a rock and the young ones can grow. Sometimes budding through the bud that I showed, or gemule. Gemule normally will happen with the freshwater sponges. So that is also possible. And very important climate change studies, sponges are helping because of their plastic growth forms. They are very sturdy in and they can adapt to extreme environmental changes. So that's why they can be very good ecological indicators. And for example, when there is a big wave or like a constant disturbance happening, they are very calm, and these guys will change their shape and they don't complain, and then they change their morphology into a different things. So it's a highly plastic animal where we can do a wonderful research, and that's why they are a good biological indicator for climate change studies. When you see what they host, most of them, they have 40 to 70% of microorganisms. So there is a high amount of microbiome. When we see it, it is just the same as our gut microbiome. That's why the microbial studies are very, very important in sponges for many research also. So these are some of the, that's why I'm telling about the simplicity and their significance. They are best hotels underwater because we need to learn what their art of living together. They host all these different guys of different groups of isopods, amphipods, polychaetes, forams, shrimps, everything. So all these guys live inside the sponge. Because of that, they are pore bearers. Through the pores, easily these guys can come inside. And why they come? Mostly for feeding or spawning. If they can hide from the predators, they can play around. They are hunting, play with different groups of animals, and they can lay eggs inside. So it's like a safe habitat for them. So that's why they don't charge anything. They are the best hotels, we say. So many things we need to learn from these guys, and what functional roles they have. They are highly good in benthopelagic coupling and nutrient recycling, because since they are constantly filter feeding, what happens is the water will be purified all the time, and the top layer of planktons or other animals will come down, and all the fishes can have the food easily because of these guys. So they are always very good in doing all these functional roles. So because of all these filtering nature, and they are excellent pharmacists. They are wonder drugs with all these interesting anti-inflammatory, anti-arthritic, antibacterial, anti-malarial, fungal, very important, anti-cancer. And even HIV and many other medicines are already being treated with these medicines. They are already in the market also. And I'll get into this bioprospecting a little while, but I'm just showing through all these importance of these sponges, what they can do. So this bioactive compounds and drug discovery is a widely important research and study. And the drugs from the sea is a big projects. Even our government is supporting the deep sea mission, and many other projects are happening. And this is in the recent World Ocean Science Congress in Goa. It happened, and this is from NIOT, so some National Institute of Ocean Technology. They are doing wonderful research on drug discovery. It means the NIO and many other CSIR institutes are getting into this study. So what we get from sponges, the biomaterials are, first thing is sponge biosilica. So biosilica, because there are siliceous sponges, so it will be highly, I'll show you what these siliceous sponges looks like in a while, but before that we'll see what are the importance of that. So they are good in bone replacement therapy. And this collagens, as I said, very important. And this collagens, we always now in Amazon and many other things, we buy them. And from the marine, from seaweeds and many other things, we get collagen as a regenerative medicine. And chitosan is very good in absorbent and wound dressing and all. And bioactive compounds, in addition, as I said about anti-malarial, terpenes are already in the market. And very good in cell adhesion. So biomedical engineering, these sponges are highly helpful. And all these different pictures that you're seeing is the sponge skeleton. So the biomimicry, we learn sponge with their skeleton. We create artificial structuring and engineering. We are learning a lot of studies from sponges, because what makes sponges so strong. And there are so many interesting things to read. I'm not going into the detail, but we are into research on oil and water separation as filters through these silica nanoparticles, and a biofilm. We are trying to use them as to replace plastics. So with the diversity and classification, we'll quickly go into them. It's not like a complete biological jargon. I'm not going into that, but you need to know what are the different groups of sponges. So sponges can be classified into 4 different groups. The first one is demosponges. So demosponges are highly siliceous. So they have silica. Then calcareous sponges, mostly calcium, that is limestone. Then glass sponges, which are deep sea, they are also siliceous. And encrusting sponges, it's a recently found different group that is coming into homoscleromorpha. I'll show you how the demosponges looks like. They are the come in different. So these are the common and most widely prevalent groups. And the siliceous sponges that we see, they have spicules like this. So if you see these important things, they are the identification guys. So with this, we will sort it out as demosponges or calcareous sponges. So I'll show you the spicules. The spicules, we call them as skeleton. These are the different types of skeleton that gives the strength to this simple guys. So calcareous sponges are very important for getting into the different groups, because here you can see a single cavity inhalant canal. That is, this canal system is different in calcareous, 3 different types of asconoid, syconoid, and leuconoid. You can see this variation slightly, like how the water and the pores come in, because when the water comes through the ostium, and this has, the leuconoid has very less cavity inside. That is the speciality with calcareous sponges. When you see calcareous sponges, see the difference in the spicules. So the varied spicule categories are completely different when you see from the siliceous sponges. And hexactinal, or the glass sponges. So the glass sponges are always very beautiful. I should tell you, they are called as the Venus flower basket. So they are glass sponges. The way it has been arranged, that is why the bioengineering and everybody are getting into research to check what kind of methodology, how they are the skeleton is being interconnected, how we can incorporate in our research. That kind of biomimicry is kind of research that we do, and we learn from these guys. Okay, so this Homoscleromorpha is a small group where these guys are mostly encrusting, and sometimes they don't have a proper, or they have large spicules. So the glass sponges spicules, the skeleton, see how beautiful they are. They are just like the flower, spicules. They are very, very long spicules. They'll be very long, but highly beautiful ones. So the encrusting, you can see the spicule category. They are completely different, but they are highly broader and bigger version. And the deep sea sponges, they look like real cotton candies. Okay, so they have a long stalk, and even with ROV and many other AUV vehicles, we can see that these guys always will be moving very slowly like this. So we found there are carnivorous sponges also in a very nutrient poor environment. These are interesting groups. They eat other sponges, and they eat other planktons and zooplanktons and all, and highly Atlantic and Arctic are explored much, and mostly these guys will be in 800 to 4,900 meters. So far, only 150 species are recorded, and here in India, we need to really explore these guys. So when you talk about sponge phylogeny, within them, always siliceous groups, both demosponges and the glass sponges always group together, while the siliceous and the calcium always get into the primitive group as a separate clade. So with all this, we know that these sponges are highly complex animals with no complex characters, with a common ancestor. So we could see that without any kind of special organs, these guys are having interesting groups. So with diversity and distribution of these sponges, we need to know that about 9,335 species are recorded worldwide. And the recorded means they are described worldwide, but there are 25 to 26,000 or 30,000 species are there yet to be, discovered. So many species, each and every day you will come into the new species and you can see. So if you want to know about a species, whether it is valid or anything, you can go to this World Porifera database, or if you want to check any kind of all the rest of the animals, you can get into World Register of Marine Species. You can note down these two websites. If maybe you guys will be knowing, but if anybody know about the species or want to check the spellings or anything, you can get into this. And in India, we have only recorded 650 species. But so for me, as a teacher and taxonomist, both, sometimes I go into the water, do motile, and sometimes many sessions like I have to stay under the microscope and check these animals. So with exploring areas, I have received samples from Gulf of Kutch to Odisha, but these Odisha and Andaman region are yet to be discovered a lot, explained and surveyed a lot. So we need to we have a lot of possibilities. So when we get into this lessons of simplicity from resilience and how they adapt to this changing world, we need to study. The first thing is their simple nature of hosting many animals and yet nicely getting into this extreme environmental conditions, they adapt. And even if the temperature rise or sea level rise, or if the temperature goes high, they try to stay calm and they survive. I'll show you some pictures, and these are some of the mm, importance, resilience that we need to study from this sponges. They are, what are the significance when you dive, and for tourists or any people, these sponges are a perfect artist of the ocean. They come in varied shape and different size, and interesting to learn each and everything. And they are great competitors. Now, the research shows that with hard corals, sponges are dominating in many places. They are dominating, and they grow even in a hard coral. They can sit on top of it and start growing very fast. So compared to hard corals, these guys grow faster. They are very strong survivors, even in murky water. Even because in Mumbai, I have shown this picture, from the household waste and industrial waste, everything comes to the easily. Even when you go there, you can see the water, sewage coming into the ocean straight away. But they don't bother. They still go and survive nicely there. And they are very strong defenders. They have chemicals and other toxins to repel against many animals. This is a holothurian, and it is a sea cucumber, and they try to bother these sponges, and it tries to release the toxins. And these toxins are important for study, important for biomedical properties and many other things. So that's why each and every sponge has some unique nature, and they are very, very friendly supporters for many other things, as I have explained. And this hard coral and sponge interactions you can see with the different shapes also. And then with the ecological role, they are habitat and also food for many fish, and particularly this turtles. Turtles love this sponge. So they are, and also, like, sometimes if some of them comes near, they try to, as the sea cucumbers come, they will try to get rid of them by ejecting the toxins and other things. And they are good with nudibranchs also. So nudibranchs are commonly called as sea slugs, and you can see how they three of them live in harmony: the hard corals, the sponge, and the phenobris, this nudibranch. So they are associated with each and every group of animal. That's the interesting thing that I'm showing. They are good with zoosandale also. This is the tennis ball sponge. And then good with mollusk, a lot of mollusk. That's also an interesting group of study. People always study their associations mostly. And this is again in Marine Drive. You can see it is there growing along with gorgonians, the octocorals. Okay, so they are again in a small rock. You can see how many species are there. So it's like a competition, and they try to grow anywhere. They have an association with the echinoderms also. And so the collection always in the deep sea is very important, because as when I showed about that cotton candy thing, because when you collect samples, you need to be very careful without disturbing them. So some common sponges from our area, I'll quickly go through, and then you will get an idea what are the sponges that we can get. So these are some of the common ones, like barrel sponge, waste sponge, massive sponge, and all. And this Tamil Nadu and Kerala coast, we need to keep in mind, because of a lot of activities, especially when this harbor activities and so many dredging and anything can happen. The water will be a bit murky. So when you dive, you need to be careful, and you then only you won't get any very good pictures. And these Gulf of Mannar always have very good, beautiful colored sponges, and these are during the low tide areas. These hard corals, even the acroporids, are highly visible. And along with that, you can see the sponge. And these are intertidal sponges from the Gulf of Kutch. And you can see how nicely they are being named, because taxonomists have the privilege to name them as halva sponge and purple sponge, dark sponge, volcano sponge. So they come in different groups. And this marine drive and whirly sponge, these sponges can grow along in a rocky bottom, and also they can come on sides. And you can see this noodle sponges, they try to branch, and with all these fragmentation, they try to spawn and grow nicely. And in Kunkeshwar and in Maharashtra, even in the rocky pools, like small pools, they survive. Even when with all these tourists happening and a lot of people there and a lot of activities happen, they are fine. Please, Goa, you can see 7 meters, 10 meters, you can see beautiful colored sponges. This is again in intertidal region of Goa. No water, nothing, and still even when the waves are hitting, still the sponges are live, and they are growing very nicely. And this is the of course Lakshadweep is a bit pristine. Water we can have a visibility of 25 to 30 meters, and these sponges are doing really good with different colors. Okay. And when I go to the other countries, Australian sponges, you can see this Bob Marley sponge with the the flower shaped sponge. All these are in spongemaps.org. You can have a detailed thing about all these sponge variations. You can see, I'm just showing you to see the differences that we have in other countries, and the Caribbean and the Indo-Pacific, the variations, just to see the difference. So why we need to study sponges? Okay, so since I'm a taxonomist, you need to, taxonomy means classification, so you need to name each and every sponge, because all these different biomedical properties, so many biomaterials, biomimicry, everything happening, and a lot of microbial, immunological work, toxicology work, everything happening, but they don't know what the species names. So that is a pathetic situation. So we need to name the sponge, we need to study them, but it is very tedious. So I know that very, very few people will be enthusiastic to sit and work very calm, and because everybody are in a rush. So now so this is the main reason in India that we are having a scarcity of sponge taxonomists, and also we have a very limited knowledge of spatial and geographical information. Very difficult to reach remote habitat, hard to reach any deposited specimens also. So that in addition to that, we have technical challenges, and even morphology differences. Some people think that when they see the picture, they think, oh, this is this group, and they start confirming and they start writing. But when you check the skeleton, it may be completely different. That's why we need to be very careful in naming them by only seeing the skeletal structures and then get into the. And of course, we need a lot of fun, and we have, of course, a lot of taxa. And as I said, we have lessons of taxonomists. So always, Bill Bryson has mentioned that taxonomy is described sometimes as a science, sometimes as an art, but it is really a battleground. So you need to be very calm and quiet and passionate about these groups. And I have been in this field since 1997, and I was always in love with this animal. So what is the recent focus of this research? So now scientists have found that, because we know that microplastics are a bit of a serious issue that we need to study, and they found the artificial sponges can get hold of all these microplastics. So they are thinking and making a research how how to make an eco-friendly methodology to farm, and we can use it widely with this to avoid and to grab these microplastics. And researchers again in New Zealand found sea sponges, the live sponges, also hold microplastics in that. So in India, that is the major interesting research that is put forward again. And as I have mentioned about this biomedical properties in them, we need to avoid overharvesting of the natural populations, because from natural population we can get only a very limited amount of bioactive compound, and we need to kill a massive biomass of sponges. So basically what they do, they will create chemical compounds artificially in the after knowing the chemical compound, they synthesize other materials. So we have a challenge, and we need to culture these groups for since they have all these pharmaceutical and cytotoxic importance. And all these several compounds we need to keep in mind that can be associated with microbes. So we need to create the farming and to support the complex microbiome also. So that is a bit of a challenge, but there are success stories. So these are spongy culture that happened in Costa Rica and Zanzibar, where they, in the shallow areas, how people are telling that these sponges have changed my life. They have given a new alternate livelihood for them. So these ladies are farming, sponge farming, and taken this sponge farming as a different profession. So how you can help in sponge research? Whenever you go along the coast, or when you are going into the sea, or whenever you see along the shore also, if you get any sponge, please get them, find them, like where you collected, which location, what is the appearance, what is the color. Sometimes the fishermen, they get these in the net, in the fishing net, they used to get them, and they just throw them off because they don't know about that. So if you are any interested, please get from different locations wherever you guys are watching, or you can collect it, and if you're really interested, we can see them. And also we need to note down where it is being collected, what is the appearance, which substratum, if you know them, otherwise, and also who collected them. So all these informations are important, and if possible, if you can freeze it and bring it to the research lab, we can store them in 99% alcohol, and then it will be helpful for molecular studies. So this is just an information, like if anybody are interested, I'm just giving through. So to wind up, there are many interesting books to read on. So these two, Systema porifera is the Bible that we say. There are so many materials are now available online from different locations, but I'm just preparing one for India, which is like a Herculean task, with putting together all these sponges with the taxonomy. It's like a one-man show. So I need a lot of hands. So with this platform, I take it as an opportunity to tell that if anybody fell in love with this totally unsung heroes of our ocean, I'll be happy to help, and then we can get into more further research and help you out to know about, or any kind of research, if you want to get into any kind of other topics for research also, because I don't know what is your background, that how many people, how many students are there, but I will be always happy to help you out, and that's why I'll leave my email with you. And with that, I stop my presentation, I think, with 15 minutes. So I'll now take questions if you have any questions. Thank you, guys. Is there any questions, or anyone wants to give any of your thoughts?
Hello, ma'am.
Yeah.
No. Why sponges are different color?
Good evening, ma'am. This is
Yes, Giri, freshwater sponges are in Kerala also. I saw the question.
Ma'am, why sponges are different color?
They have different pigmentations. So they have purple, red, yellow, orange. So many different pigmentations are there. So that is a different wide topic. So each and every sponge has some unique characteristics and pigments, just like hardcores. And also this microbes are there.
Okay, I thought like corals are attached with sea anemones because of sea
No, no.
anemones.
No,
Okay,
no.
okay.
Because these, as I showed, they have a different association with different groups of other animals also, like echinoderms, sea anemones. Everywhere they get associated easily. They are the very simple, humble groups that I said.
Yes,
Simplicity,
ma'am.
and at the same time, they are highly important for all these, research that I have mentioned.
So, ma'am, which pigment are present in their cells?
Which one? Sorry.
Which pigments present in their cells?
Which pigments?
Present in their cells because they are multicellular organism, na?
Yes, yes.
Which pigments found in their cells?
Terpenes. There are different research happening. Maybe I can share some articles about in detail, but from in India, I think very few of them, getting into research on that.
Why they, ma'am, during evolution process, why they are bearing different? What is the significance? Why they have different pigments?
Uh, why they have different pigments? Um, so each, because the pigment research I haven't done, but all these, for example, mostly I think with the microbes or with the attracting other animals, um, for as I said, with this polychaetes, amphipods, many other groups are there. So we actually don't know who is contributing to these pigments as such, even with whether it can be a microbes or that research is still there.
Exactly. That's why first I asked question why different color. I thought
Yeah,
like it's the coral association with the sea anemones that because of that. But
because
you told
corals we can always say it is with algae, right? So and also corals also have like acroporids, stylophores, all these have different colors, right? Only when the algae is gone, they will be completely bleached, and that's why you can see. So sometimes this sponges, the interesting thing is some sponges with all these purple colored and the yellow colored one, they will be bright colored underwater. When you take them and put it in alcohol, all these pigments will be coming into this ethanol easily, and the color will be totally lost, and you can see very gray colored or off white color. Suddenly it can change. So that separate pigmentation study is a wider topic, I think. Sorry, I haven't gone into that.
okay, ma'am. That's okay. Ma'am, which category of the poriferan species sexual reproduction develop during evolution process? In which group of poriferan species?
Um, because most of the demo sponges, as I have shown them, get into this sexual reproduction, and certain groups like because should I go into the slides and show the species, or
Okay, ma'am, no issue.
are you interested to know which species or which stage?
Which species
Ah,
during the
well,
evolution?
because a lot of demo sponges, calcareous sponges, because even in certain group, they will have both sexual and asexual reproduction within them. So I cannot completely say that calcareous or demo sponges or glass sponges will have sexual reproduction.
How they develop, madam? How they develop this mechanism?
They are they have hermaphrodite cells inside, like both eggs and sperm inside them. So the fertilization actually happens inside, and then the larvae will come out and then fall in. So the larvae will be motile for some time, and then they fall into a proper substrate.
Ma'am, same animal, how the meiosis is going on? How the gametes formed?
How? What is the question?
In same animal, how the meiosis process, how gamete forms?
This embryology is again a vast topic. Inside them, when they grow itself, they will get that eggs and sperms in them at a certain levels. But as I said, these are simple groups. They don't have a proper reproductive system, proper circulatory system, proper nervous system. Nothing is there. But it is a cellular grade of tissue organization. So naturally, when the cells are being formed, after a certain in a maturation stage, immediately this eggs and sperms, just like easily they and the spawning season can happen in a I can show the video nicely, because this sponges can push out this sperms nicely, like a fumes, black white fumes. So even when the favorable condition, ecological condition arrives, it can happen easily. So in the other interesting thing, just like hard corals, when you take two different species, the sperm and the eggs, they can join with only that particular species. So that is the interesting thing that make the scientists so curious to study about this sponges, which we don't know actually how this things happen. So many interesting questions. Good.
Good evening, ma'am.
Mm.
My name is Priyanka. I'm from Calcutta in West Bengal. Thank you for a wonderful session, ma'am. But I have a couple of things to share. So, a couple of friends and I, we have a marine biologist with us. We're planning, we have been looking into the coastline along Bengal, Odisha, and parts of Andhra also. But specifically in Bengal and Odisha, I haven't found any sponges along the coast, so to speak. And we have a ton of beaches along Bengal and Odisha. In parts of Odisha, if we get into the depths, yes, but along the coast, not really. So, is there a particular reason why this is happening, or do you think there's, because the rivers are draining, so there's a lot of alluvial there, clay deposits? Do you think it's because of that?
Very, very good question, Priyanka, because this is from Odisha. We have reports of sponges a long time ago during expeditions like in 1900s, 1930, 1931. There are few records, because after that, as you mentioned, there is the shallow region. We haven't seen any new records, because this is due to developments that has been happening, and their habitat would be totally been destroyed. That may be one of the reasons.
Right.
But the thing is, as I mentioned, they survive in highly challenging environment. They try to be there, but the problem is, if that case is there, and even with the olive ridley nesting patterns and everything, that has also been changed a lot in Odisha, right?
Yes,
Yeah.
yes.
So I think mostly it has to, because I haven't explored in the subtidal regions, but
Okay.
if you can collect, if you can,
Yes, ma'am,
get
for
it,
sure, for
if
sure.
you,
Is there any way I can get in touch with you and stay in touch? Because we'll be going right down the East Coast and we're concluding this in Varkala. So it's a long trip. We'll be walking, beach combing, collecting samples, and going right up to Varkala. So I would definitely want to be in touch with you if I can be of any help. If all of our efforts, we'll be diving as well.
very good, very good. Is it your project or a PhD project or
Yes,
something?
ma'am.
Yes.
No, ma'am. It's a self-initiated project because I'm basically a wildlife photographer, but we have a couple of researchers in the team also. So, we realized in a very awkward way that a lot of documentation of mammals and birds is happening, but marine life is completely neglected.
Yes.
So,
Yes.
so all of us are going to the national parks in the mainland, but no one's going to the islands, no one's going to the marine national parks. So we wanted to collect data, uh, complement research, and also bring this to the forefront to the masses. Because if they don't know what's getting destroyed, then they can't conserve it. So it's an independent effort.
Yeah. So that's interesting one, Priyanka. But I will be really happy. I really want the youngsters to join, because we need to form a real good team to get into
Yes,
this,
ma'am.
go
Yes,
save
ma'am.
this, because as you said, that has been highly neglected, and
Yes,
the
ma'am.
people coming into this and studying is very less. So that has been important, and I'm happy that you guys are forming a good team and getting into this. I will be very helpful, and I can collaborate with any kind of projects or
Sure,
anything that
ma'am.
you come across.
If you
Good.
could just share your email, then I'll get in touch with you.
Yes. Did you noted the email? Because I just stopped sharing the screen.
No, ma'am.
Okay.
Is it in call messages or?
I can put it in the chat. Okay.
Okay, ma'am. Thank you so much. Thank you.
Okay.
I have a comment and a question.
Yes, please.
The comment is that, first of all, thank you for that interesting introduction to a loving group of organisms. I personally feel that they are very successful. They have
Yes.
so successfully navigated the 5 major extinctions on the planet, had been living in this planet much more time than human beings. And as per the second law of thermodynamics, it says that simpler organisms are much more successful than complex organisms like human beings. Like, for example, a simple block in the artery leading to heart can kill a human being, but you can
Yeah.
never do that with a sponge. They survive in whatever form they have. So basically, simplicity is a big successful thing for them, making them adaptable, flexible, and able to survive for a long period of time. And the question is that, can you, based on your experience, could you list out what are the problems which sponges are facing to survive on this planet, especially since you mentioned about the port making process. In Virginia, what has happened is that the entire fauna in the seabed has gone. So, could you just list out the challenges faced by sponges?
See, as you have in your question itself, the answer is there. The anthropogenic activities are the first important thing, even though they are good in filtering and with all these sedimentation. When it happened, we try to trigger that entire the area with all these dredging and so many other activities. So as you know, with the Birmingham harbor or any, I'm not telling into specific things, but any activities that has been happening along the coast, it's a threat. Like even as I showed in Mumbai and many other, the sewage, the river runoff, the freshwater intrusion, is always a threat, because they need an ideal, temperature and conditions to grow, even though they filter all the time. So if the sediment size or the particle size grows bigger, what will happen? So completely, because the ostia is very small, and at the same time some species like the tennis ball and many other species, they have a little bit of a small broader one, but still there is a limit to push this guys a bit. So with the temperature, that is the apart from the natural causes, anthropogenic is an important threat. And also human intrusions, not only with dredging and many other things, like tourism activities. When we do all these touristic, even in Mumbai, Maharashtra, and many other places, even in Lakshadweep, I have seen tourists, just for sake of one picture, they just smash and kill the whole sponges in one go. Even they do that with hard corals also, which they built millions and millions of years to form the whole massive corals with that single polyp. So first thing is anthropogenic activity is really a threat, that I will say. Apart from that, temperature rise. But in certain places, these El Nino and La Nino activities are a bit not a very big threat. That has been, research says that half of them, they survive even in extreme temperatures, but some of them, they die off easily when the temperature is so high. And apart from that, any kind of developments, I will say, is a major threat. And ecological conditions, when it changes, they will stop growing, but still they are trying to go through and survive. But that's a very good question, sir.
Thank you, Dr. Anita.
Any other questions?
No, ma'am.
Yeah.
Yes.
Okay, we can.
Ma'am, one thing I want to share one information. Actually, I'm from basically from Gahirmatha area, the mass rookery nesting of Oliver sea turtle.
Yes, yes.
As being a wildlife ecologist, I have updated my 6 seniors of PhD completed, one in JDSI, Pune. Anyway, one from Dr. Sajan Jan from Kerala. So they have studied so many sponges species found because they have got analysis and they found sponge provide the major diets. So many sponges found in Odisha. There is no any mass rookery shifting the Arevada. There is no disturbance. One thing is Gahirmatya area little bit disturbed because of the Paravati port near. Another is Wheeler Island, where is the all missiles launching testing, everything going on. The ISRO's near Chandipur, Baleshwar district. So that's why it's little disturbed. Otherwise, in Rusikulia in Ganjam district, South Odisha, one also guy from P. Muralidhar from Tamil Nadu, he also studied through WI, MSc dissertation. So there is no much disturbance in Arivada or Mass Rookery because
So,
of sponge.
so even in Aripara, during in that area, could you see even in the shallow intertidal region a lot of sponges?
Yes. My home place is near just 10 kilometers from
Okay,
sea
okay.
coast. Yes.
So those places, I think, even like the reports and the records so far there, many of the sponges needs to be revised to the latest,
Yeah.
things.
Paradip port is, yes, Paradip port is very near to us because of this heavy pollution. So many species vanish. Now, during my childhood, the that sea beach, sandy sea beach become white sandy. Nowadays, pura fully blackish. So many fauna species also died. And molluscans also, earlier, so many molluscan deposited in the sandy beach. Nowadays, you will not find this red crab also, so many crab species, arthropoda, so many. Obviously, disturbed, but not in other areas because of this paradip port only. Because oil splitting, so many things there.
Yeah, that's a interesting place to research and study. To with the reduction in the beach, and also whether it is favorable for the turtles to come and go, and for these sponges also. Okay.
Yeah, yeah, no, turtles are not coming to these areas to where destroy is there. The sporadic nesting going on. But I'm talking about in Resipili and Debi River mouth in Jagas, that mass rupee going very nicely, and Resipili nowadays population doing more numbers of individuals that are mass nesting. But
Okay.
Gahirmat a little bit disturbed because of this launching pad, this Wheeler Island, and also that called
Thank you.
Abdul
Thank
Island.
you.
Yes, yes,
Thank you, Amita, for the interesting information. Yeah.
Ma'am, there is a question in the chat box asking uh, any molecular study of anti-cancer property, like which protein is targeted in cancer cell and not. So asked by Chandana Vishwakarma.
Okay. So that is interesting. Another interesting thing which I didn't go into detail. Only important thing in New Zealand and many other places, they have found the real right protein for cancer research. And the interesting why they target sponges, because generally we do chemotherapy, and we know that the good cells also will be affected, right, in normal chemotherapy. But with this sponge anti-cancer property and the biomedical the compound from sponges, it will target only the cancer cells. That is why the sponge molecules are very highly targeted for anti-cancer research. And in India, we are yet to find a lot of specific sponges with anti-cancer. But in NIO, few sponges are already been reported with anti-cancer property, and we found the protein, but yet to produce and get into the further research on that. So we have found certain groups of sponges already having cancer properties in India. We have spotted that, but we haven't gone into the next level of research on testing them medically and getting them into the market. Good question. Any other question? I think.
Yes, ma'am, one more.
Yeah.
She's asking, is there any symbiotic association and nitrogen fixation like algae?
So I saw the question. This is that's what I have shown. The symbiotic association is always with very algae are there. Even the tennis ball sponge that I have shown, that is Cinecarella, they have a very good association with algae, and the nitrogen fixation has been happening, and so many research are there. But the thing, since I'm doing only on the taxonomy, but I do the interdisciplinary parts with this biomedical properties to help in different microbial and other social. And the symbiotic thing, I have done a research not in India but in the Great Barrier Reef. So it is there. There is a high symbiotic association with algae is always there. Kavya, thank you for the question.
Does anyone have any more questions?
And if there is any questions, or if you want to communicate or tell or ask any questions, also feel free to use my email, and you can get to me, and I will be happy to, because any kind of questions are welcome. It's a interesting thing for me to learn many things also. Thank you. So I hope you have the email with you. So yeah.
Yeah, thank you, mom. Good night, mom.
Thank you so much. Thank you. Wish you all the very best.
Thank you, mom.
Bye. Good night.
Thank you, ma'am, for this session. It was deepened our understanding of marine biodiversity. And we also thank everyone who joined and actively participated. Before we conclude, we are excited to share what's coming next. Tomorrow marks a major milestone for Biorix as we officially launch Biorix Bio Talks, the student knowledge sharing series with its inauguration by Dr. T.V. Sajiv, Chief Scientist at KFRI. This will be followed by our first student lead session on invasive alien species, threats to biodiversity and ecosystem by Alisha Anil, scheduled on April 5, 2026 from 7:30 to 8:30 p.m. via Google Meet. With that, we officially conclude today's session. Thank you all for being a part of this discussion, and we look forward to seeing you tomorrow at the BioTalk inauguration. Thank you.
Best wishes for your program tomorrow, Sonia, and to all, and Sajiv sir. Thank you.